So good afternoon, everybody. It's a pleasure to be here. I just want to thank PacBio for the invitation to speak today. So as mentioned, I'm going to be talking about our experience with long-read HiFi sequencing at Stanford, but also at the institution that I was prior to joining Stanford in 2020, Mount Sinai in New York. And so for an outline of my talk today, I'm going to introduce briefly the Clinical Genomics Laboratory at Stanford, and then walk through our experience with HiFi sequencing applications and splitting it up into Amplicon sequencing, enrichment panel, HiFi long-read sequencing, and HiFi genome sequencing, and then ending off with our experience with actually implementing a clinical pharmacogenomics program with a HiFi sequencing. So the Clinical Genomics Laboratory at Stanford was actually set up back in 2015 as an agreement between both the adult hospital and the children's hospital to provide the infrastructure for clinical germline NGS testing, as well as a scalable computational infrastructure, as well as interpretation team and pipeline development. And I really want to acknowledge you and Ashley and Luann Hudgens for really spearheading the development and the initiation of the service. And so initially, the genomics program launched an enrichment-based exome in 2018, which was sent out to Personalis, targeted variant detection as well as reanalysis. And so it was a pleasure to join the laboratory in 2020 because the idea was to leverage the infrastructure that we have for genome-based diagnostic testing. And so what we have available to us now is genotyping with the GINA, NovaSeq sequencing, as well as a robust computational environment for our pipelines and data processing and storage. And we were really thrilled about 2 years ago that Stanford Healthcare invested in a Sequel IIe for us in the Department of Pathology in the clinical laboratory environment. Unfortunately, this came out just a few months before the Revio, but we'll talk more about that a little bit later. And then we also have rapid point-of-care genotyping available for some pharmacogenomic targets. And so in terms of what we've been focusing on since 2020, we validated short-read genome sequencing for diagnostic testing of inherited cardiovascular disease diagnostic panels in 2021. We got rid of the Personalis exome that same year and developed a custom exome on a genome backbone in 2021. And just to go through, we also partnered with nephrology in 2022 and then preventative cardiology in 2023 to really drive genome-based diagnostic testing, where we bioinformatically strictly look at the clinical content that was indicated for that test. And then the extra genome data goes back into our translational research programs where we look for gene discovery. However, for today, I really wanted to focus on, of course, what we're doing with HiFi genome sequencing or HiFi sequencing in general and our experience with Amplicon enrichment and genome sequencing. So with Amplicon sequencing, given our interest in pharmacogenomics, we first wanted to leverage long-read sequencing for some key pharmacogenomic genes that are very difficult to interrogate by short-read sequencing. And the setup is relatively straightforward, where we just do long-range PCR, we barcode, and then we subject to HiFi sequencing. And of course, we get the really beautiful variant calling and complete haplotype phasing, which is really important for pharmacogenomics. And one of the most important pharmacogenomics gene is CYP2D6. So this was our first target that we selected to leverage this method with PacBio sequencing. And if we compare it to short-read sequencing, IGV looks like this, where we have CYP2D6 on the left, and the two pseudogenes, 2D7 and 2D8, are on the right. And so this is an example of off-target sequencing, where those short reads should not really be piling up there. They really should be focused on the gene itself. But when we compare this to long-read PacBio sequencing, we can appreciate that the long reads really specifically interrogate only the coding gene and completely avoid the pseudogenes for much more accurate variant calling and haplotype prediction. We've used this technique now on a couple of other pharmacogenomic targets. SLCA6A4 is a gene that's implicated in antidepressant response. The main variant that drives that response difference is a promoter, variable number of tandem repeats. There's a short version and a long version, but it's highly repetitive. This is an example of IGV looking at the short reads, but that can't even align to the promoter given the repetitive nature of the region. But again, if we look at HiFi reads across this promoter, we very beautifully can completely sequence across the 2 kb repetitive promoter. And in this case, this is an example of a heterozygote long and short allele sample for this gene in this promoter. More recently, we published an Amplicon strategy for NUD15. So this is a hot pharmacogenomic gene that was discovered a few years ago by Jun Yang at St. Jude's for thiopurine toxicity for children with leukemia. So the classic gene was TPMT, but there was still a subpopulation of patients that were experiencing life-threatening toxicity with the treatment. And June discovered NUD15 variants as germline variant predictors of toxicity. So this very quickly became clinically actionable into CPIC guidelines as well as PharmVar star allele categories or classifications for the star alleles, haplotypes. But also this gene, like CYP2D6, is not very big. It's only 9 kb. So this allows us to use that same Amplicon strategy where we can actually amplify the entire gene, barcode it, sequence it, and phase the haplotypes. And so this is just one example of a Coriell sample. The short read sequencing called it a star 1, star 2, which is one normal copy and one loss of function copy, which is an intermediate metabolizer. But when we looked at the long reads shown here, I'm not sure if this has a... But anyways, on the top two is the short reads that are unphased, whereas in the middle, this is the phased diplotype with HiFi sequencing. And when we zoom in on the bottom, there's a very complex insertion and a deletion on the two different alleles, which the short reads missed. So the actual diplotype was star 2, star 9, which is a poor metabolizer. So it actually changed the clinical significance of the call when we moved from short read sequencing to long read sequencing. And this is just another example on the right, comparing the short reads on the top that certainly can sequence across the gene, but they cannot phase the haplotypes compared to the long reads on the bottom, where you beautifully phase across the entire gene for full gene architecture. So that's a quick overview of our Amplicon experience. Now I want to pivot to our enrichment panel strategy. So since joining Stanford in 2020, I've been collaborating with PacBio as well as Twist Bioscience. And Fritz is probably going to talk about this as well after this talk, about designing a alliance panel for pharmacogenomics. So we collaboratively put together a gene list of high actionable, high evidence CPIC genes that are underlined, that are in underlined, and then full gene coverage, which is in bold, as well as other additional content that's more, maybe not be clinically actionable today, but it has emerging evidence for validity and utility, and maybe more prone for research content. And so this has been a really exciting collaboration, and Tina Han is here. So I want to thank her and her team as well for helping us optimize the panel, as well as PacBio for doing a lot of the sequencing in Menlo Park as we optimize the panel. And so just to give a snapshot, we're getting really good coverage per gene across all 49 or 50 genes. We also include the mitochondrial genome as well. Average read length is 7 kb. We do 24 samples per sequel to SMART cell. So of course, with the Revio, you can multiplex many more samples than what we're doing with the sequel. And just to give you an idea of what that looks like, I mentioned before the SLC6A4 gene promoter. On the right is just all 24 samples showing really beautiful full gene coverage with our HiFi enrichment. I mentioned the mitochondrial genome. We can also sequence the mitochondrial circular genome in the same reaction. And then, of course, CYP2D6, which is a very important pharmacogene. Again, we get really nice full gene coverage using enrichment as opposed to the Amplicon strategy. But one of the big challenges just to give a few more examples of what this looked like and underscoring the phasing. So on the top left, this is TPMT. This is a single sample, so you can appreciate that with the two colors, red and purple, these are the two phased haplotypes across the entire gene, which is really powerful for haplotype and diplotype prediction. Some of our targets are single variants, OPRD1, which is shown here on the top right, really beautiful coverage. And on the bottom, some of these difficult genes are the CYP450s. I mentioned CYP2D6 already, but this is CYP2B6, which is also structurally very variable. But again, on the bottom, we can appreciate really clear, full gene phased sequence coverage, haplotype and diplotype prediction. One of the challenges, though, is actually going from the variant calling to those star allele predictions. So I'll mention this a little bit later when we talk about pharmacogenomic implementation. But thankfully, there's a lot of tools available now that are algorithms that, based on the VCF or based on the BAM for the structural variants, they can actually infer or predict which star allele the sample has. So we don't have to do it manually anymore. Because we've, at this point, with high throughput sequencing and with the phased full gene HiFi coverage, it's no longer feasible to manually do that star allele translation. So thankfully, there's a host of tools available that enable us to do the star allele translation and phenotype prediction. So from the Amplicon to the enrichment and now to our experience with HiFi genome sequencing, the rationale for moving to genome, I'm sure Tommy mentioned this earlier this morning, but it's really, in my mind, the accuracy. So from a clinical diagnostic lab perspective, there's nothing more important than being able to comprehensively detect our germline variants. And so when we validate our panels or our exomes or our genomes for accuracy, this is the tables that we generate. And I can appreciate that it's probably too small for you to visualize. But just to give you a brief overview, on the left, we stratify by small variants, SNVs, indels of different sizes, and at the bottom, all indels, and at the very bottom, SNVs and indels. And then we measure recall and precision across the genome in a bottle truth set, which we've heard a lot about benchmarking today already. But in addition to doing the benchmarking, we also stratify it by several different strata that are defined by the Genome in a Bottle or GA4GH consortia, low complexity, low mappability, segmental duplications, all difficult regions, not in any difficult regions, and all high confidence. And the reason why we do that is because it gives us a better overall predict or assessment of performance. And so we've done this a lot with our short read data sets, but it was really encouraging with our HiFi whole genome data that we're getting much greater than 99% accuracy, even in the difficult regions of the genome. And then in the non-difficult regions, we're surpassing 99.9% accuracy with small variant calling, which is really, really encouraging from the clinical laboratory perspective. So this is, as you likely know, a product of depth for many reasons. And we've also compared this to our short read genome data validation work. And so this is F1 scores of both SNV and indel accuracy so that we can appreciate that at 20x HiFi genome sequencing, we're actually getting small variant accuracy that's consistent with greater than 30x short read genome sequencing. In our diagnostic lab, we're actually pushing that to 40x for short reads, but now we know that we can actually dial that back for HiFi genomes and getting the same level of really high accuracy. Again, though, going back to those genome in a bottle stratifications, it's not just those four major strata that genome in a bottle provides. There's actually up to 100 specific bed files for all different types of regions in the genome based on GC content, tandem repeats, and a whole bunch of other MHC region, a whole bunch of other specific areas of the genome. And we just have a subset shown here, which is basically HiFi genome sequencing F1 scores on the left, compared to short read genome sequencing F1 scores on the right. And where I hope you can appreciate, and on the bottom are the different variant types, single nucleotide variants, indels of increasingly greater size. So as we move to the right of the graph for the short read accuracy results, you can see how the short read indel calling really falls off at the larger variant types compared to the HiFi genome variant types, which maintains its accuracy across these high or larger indels in these difficult regions. Really compelling for us in the diagnostic lab. So in terms of how, in addition to validating the accuracy, another major component for CAP and CLIA is reproducibility. And so we've taken our genome in a bottle sequencing data and compared it using genotype wild type genotype concordance with publicly available HiFi data from both sequel and Revio sequencing, and again stratified by genome context. And the take-home message is that between experiments, intra-run sequencing, we're surpassing 99% concordance for our small variant calling, which is again really compelling, but also very important for validation. In addition to the reproducibility and the accuracy, another major component of validating a clinical test is the specimen type. And so the most commonly used specimen types for genetic testing are blood and saliva. And so we have a battery of samples with paired specimens from different individuals for both blood and saliva, as well as cheek swabs. And again, the take-home message was really encouraging, which even though the oral samples can have high bacterial content to interfere with short-read sequencing as well as long-read sequencing, we're getting over 99% concordance between paired blood and saliva and paired blood and swab samples. So in terms of how we've kind of supported this in some of our early translational projects, I just want to mention a few projects. We have an internally funded MCHRI grant for pilot HiFi genome sequencing for kids who have already had a negative short read exome or a negative genome. And this is with Dina Madelon in medical genetics and pediatrics at Stanford. And that this is also in parallel to the Stanford Gregor, or the Gregor Stanford site, which is the NIH-funded consortium, and the PIs are Stephen Montgomery, Matt Wheeler, and John Bernstein. So all of our long-read HiFi work now, we're kind of doing in parallel from the pathology laboratory as well as the Gregor group, who are more on the translational research side. So with respect to our pilot testing, this is our cohort. We've got them all consented and actually all sequenced to date. So the majority have had previous trio negative exomes. The majority have also been tested internally. We also intentionally tried to recruit as many patients as we could that had one likely pathogenic or pathogenic variant in an autosomal recessive gene, with a hypothesis that the long reads will pick up the second variant. But these data are still under interpretation now. But just to give a snapshot of the quality, we're very excited. We've all of the quality metrics look very good. We average about 20x, about 13 kb per read length, and getting really consistent sequence identity, as well as total SNVs and total indels are consistent as well. So in terms of our workflows, then, to summarize, we have the clinical short read whole genome workflow that's in production. So now, as I just mentioned, we're in development of our PacBio HiFi genome workflow. So we're leveraging all of the best practices from PacBio Deep Variant, PBSV. We're also bringing in the target tool as well as HiFase, as well as the methylation data we're now trying to pull in. And then together we're collaborating with GenX to support the interpretation of this. So this is ongoing, but hopefully we'll have more to report on this later this year. And as I mentioned, the Gregor Consortium. And so the Sequel2B is in pathology department and Stanford Healthcare. And we're really leveraging this for these pilot projects, as well as bringing up our clinical HiFi sequencing. But at Stanford, they have recently invested in the Revio, where the larger bulk of the genome HiFi sequencing for the Gregor program is ongoing now as well. So we help them out with our workflows, and we're sharing our best practices. But moving forward, all of the undiagnosed children coming from the genomics lab who are going to have their HiFi genomes done, as well as other multi-omic assessments are going to be done through the university side with the Gregor program on the Revio. And so the last thing I wanted to mention was to circle back to the pharmacogenomics panel and our work with implementing clinical pharmacogenomics. And this is work done in collaboration with my friend and colleague, Terry Klein, who runs FARM-GKB at Stanford. And so she's been doing PGX at Stanford for 20 years, but very frustrated that the healthcare system has yet to actually implement PGX testing. So we applied to the Stanford Medicine Catalyst program to actually try to fund an implementation program driven by kind of the IT infrastructure that's needed to do pharmacogenomic testing. And so this is just a hypothetical workflow for genetic testing in general. But for PGX, what's really required is in the electronic healthcare record system to have that kind of functionality to provide real-time clinical decision support to providers with actually what to do with the PGX information, increase the dose, avoid the medication, etc. And so there's several companies that are available to actually bundle in with their service into Epic or Cerner. But the irony is that all of those companies use the information from FARM-GKB and CPIC, which Terry is the principal investigator for, which are NIH-funded PGX resources. And so this was our pitch to Stanford Catalyst, is that, ironically, if we want to do it at Stanford, we'd actually have to pay these companies. So why don't we build something internally ourselves? And so the prototype for this is a tool called FarmCat that also Terry's group has developed, which takes raw VCF data from a PGX gene list, and then it transforms it into those star alleles that I mentioned, also transforms it into a metabolizer phenotype, and then also brings in the recommendations from CPIC or the FDA and other resources. And so this is what we've been working on for the better part of 2024. And again, we go back to Twist and PacBio because the panel that we developed collaboratively, we're now bringing in to the pathology department. And so the team is actively working on doing all that analytical validation work that I mentioned before with the genome data, and also bringing in our interpretation tools like FarmCat and other automated resources. So going back to our workflows, this is now kind of our third workflow, where in addition to the short read genome, the long read genome, and now the HiFi enrichment panel workflow, we're actually validating this. This kind of jumped ahead of our genome work. We're actually validating the end-to-end architecture for getting an order through the electronic healthcare record system, doing the twist capture and HiFi sequencing in the lab. We have a more focused informatics workflow of alignment, variant calling, and phasing. But then we have an audit. Instead of using a third-party software, we're building that automated interpretation workflow that's using some tools from PacBio, Pangu, and PB Starface, as well as FarmCat that I just mentioned. And then now we're filing these results discretely in the Epic's genomic module. So that enables that real-time point of care, clinical decision support, and best practice alert advisories for a subset of medications that we're hoping to deploy in primary care later this year. So I know that's a lot in one go, but just to summarize, long-read HiFi Amplicon sequencing enables highly accurate and phased variant detection across clinically significant genes. And my focus has been on PGX genes as well as some Mendelian variants as well. The enrichment sequencing is an ideal platform for pharmacogenomic panel sequencing. I'm biased because I was involved in this project, but we're actively validating it now for clinical implementation across our healthcare system. And then finally, as you probably heard this morning from Tomi's talk, is that HiFi genome sequencing is highly accurate and has greater sensitivity across these challenging regions compared to short-read genome sequencing. So again, thanks for the opportunity to be here. I just want to thank and acknowledge everybody in the clinical laboratory, as well as our research team, our advisory committee, our colleagues at the Gregor Stanford site, FARM-GKB, the Catalyst folks, PacBio, and I should also have put Twist on here as well, too. But happy to take any questions if anybody has time. Thank you.